Howdy, this is Browdy, coming at you today with the second of two lectures for the DNA unit, or Unit 4. First time around, we talked about DNA structure and function, and today we're talking about DNA history and replication. So let's get started. So the very first thing is kind of in the late 1940s, early 1950s. The question was, what is the shape of DNA and how does it work? So DNA had been discovered at this point. People knew that DNA existed, but people didn't necessarily know that it was the genetic or the molecule of inheritance or the molecule that carries the genetic code, and people didn't necessarily know what it was or how it functioned. And so the people that we're really going to talk most about are two guys named Watson and Crick. So James Watson, right here, and Francis Crick, along with a guy named Maurice Wilkins, won the Nobel Prize, I believe in 1962, for discovering the shape of DNA. And so what they did is they built 3D models, as you can see here, using like tin and cardboard. And they did this with the help of photos by an X-ray crystallographer by the name of Rosalind Franklin, and we'll talk more about her later. And so what they are known for discovering is not DNA, but the shape of DNA, which they found to be a double helix, which is very similar to a spiral staircase shape. And so the DNA double helix, they said, had the backbone on the outside, made of sugars and phosphate, which this we've already talked about a few times, and then nitrogen bases on the inside. And so I said that I would talk more about Rosalind Franklin. Well, the first person to take a photo of not a photo of Rosalind Franklin, of DNA, was Rosalind Franklin. And she used what's called X-ray crystallography, which is basically an X-ray imaging technique. So this is Franklin, and this is one of her probably her most famous photo. And what this is it suggests that DNA is a double helix of even width. And so this is a picture looking from the top down at a DNA molecule. So it's a little hard for us to see at this point how exactly that works, but we're also not X-ray crystallographers or the biologists studying DNA. Here, basically what it's showing is that there's kind of this classical X pattern that indicates a double helix, and we can see these bands around the side that show that it is kind of an even width all the way around. And so the thing about Franklin, though, is that she didn't just kind of willingly help out, or wasn't a partner necessarily with Watson and Crick. So they actually didn't have her permission to use her photos. And so this photo is actually what suggested to Watson and Crick that DNA was a double helix. She just didn't know they got it from her. And so she worked in a lab that was run by this Wilkins guy, Maurice Wilkins. She had shown Wilkins, because he was her boss, he had shown her she had shown him her work in her pictures, which were really impressive and really helpful. And Wilkins was then, unbeknownst to Franklin, passing these notes and images along to Watson and Crick. So Franklin is actually also the one who determined that the backbone of DNA was on the outside. Now, to us, we go, oh, obviously it is, but we've also always been alive when DNA has been known, right? Or the structure of DNA has been known. At the time, it wasn't. And so they were unsure, and Watson and Crick had actually thought the opposite. So Franklin had been the one to suggest to them that it was on the outside to begin with. And so to complicate things further, and really one of the main reasons I want to talk about this, is that Franklin's work wasn't incredibly well respected because she was a woman scientist in the 1950s, not to mention she was Jewish on top of that, which, the 40s through 50s was not a wonderful time for the Jewish people. And so science has long been a field that's been dominated by relatively wealthy white men. And that's not to say that those people are bad people by any means. But not everyone is great, and Watson and Crick specifically kind of took advantage of Franklin's position within the field and within kind of their social structure, and really took advantage of what she was offering, and she by and large got the short end of the stick. And in Watson's book that he published, or his autobiography that he published some years later, really described her somewhat demeaningly, calling her nothing really more than Wilkins' assistant. And you can see videos of him in TED Talks where he really downplays the role that she played. And so when Watson, Crick, and Wilkins received a Nobel Prize in '62 for discovering the structure of DNA, Franklin could not receive that, nor was she ever nominated, because she had actually died in 1958 from cancer, ovarian cancer, and we believe that that was caused by the X-ray work that she did. So at that point in time, safety measures were obviously a lot less strict than they are now. The effects of X-ray radiation were less well known. The causes of cancer were less well known. And I mean, if you imagine going to the dentist now and getting an X-ray, you wear that lead vest. Well, she worked around X-rays every day with, from what I understand, is a lot less precautions. And so, unfortunately, she died a few years before the Nobel Prize was awarded. And the Nobel Prize cannot be awarded posthumously or after someone has died, and so she could not be nominated, unfortunately. And so here we have just a little bit of a timeline of kind of some key events in DNA history, and I'm less worried about you knowing these. These are things that we would maybe talk about in a non-COVID sort of world, but since we're kind of bringing things down, cutting things down a little bit to condense the timeline, I'm not too worried about you knowing a lot of this stuff. So as long as you know Watson, Crick, and Franklin, and kind of know that relationship that they shared and how Franklin really was taken advantage of in the situation, I think that's the most important thing. And again, like I said earlier, science has been dominated by really one population for a long time, and we don't necessarily want that. Well, not necessarily. We obviously don't want that to be the case. There are a lot of people who have had great scientific ideas in the past who maybe just haven't been recognized because of the color of their skin or their gender or their ethnicity or anything like that. And that is obviously not a good thing. And so, as we're trying to diversify science, I think it's also important that where we can, we recognize people being mistreated and people who are otherwise hailed for their great discoveries. We also want to recognize when they have done wrong. So we don't just want to praise these people with disregard for all the people that they've negatively affected along the way. And so that's kind of the brief version of the somewhat dramatic history of DNA structure. So we're gonna move on now to DNA replication. And so DNA has to be able to copy itself perfectly over and over and over again in order to make sure that proteins and cells function properly. Oh, excuse me. And so part of this can go in this first box here. It's not necessarily what's meant to, but cells have to copy themselves. And so that cells and people can grow, and so that organisms are able to replace dying cells. So not just people, just all living things. And every one of your cells needs a complete set of DNA, with the exception of sperm and egg. So what we see here is a brief, kind of quick version of the cell cycle, or mitosis specifically, which is asexual cell division. So here we see a cell that has the full amount of DNA. We'll say it does. Here we see DNA replication happen, and then the cell now has twice the amount of DNA. So these X's that we see, that's how we typically see chromosomes, are actually the duplicated or the copied versions of the chromosomes. Now the cell starts to split, the DNA goes to either side, and then we end up with two cells that have the correct amount of DNA. But so replication really means kind of the same thing as cloning or duplicating or making a copy, and so, or making a replica. And so DNA needs to be copied so that the cells can grow and so that cells can be replaced as they die. So making new cells is really the purpose of DNA replication. And again, every cell needs a complete set of DNA. The reason DNA is able to make perfect copies of itself so quickly is because of the base pairing rules. The base pairing rules that make it so A can only pair with T and C can only pair with G. So that system, that structure, makes copying DNA a lot easier because there's really only one option for every base. And DNA replication is what we call semi-conservative. Now, that's not to be confused with selectively permeable. People often get those two terms confused. Remember, selectively permeable is what we call the cell membrane letting some things through and not others. Semi-conservative is a totally different thing. And so if we look at the picture here, we see a chromosome, which remember is a long piece of DNA that's coiled up really tightly. And here we see the original piece of DNA that's being copied. Now it gets split by something we'll talk about in a second, but as it gets split into these two strands, the new DNA is built as the other half of those two template strands. And so what we end up with is we start with one DNA molecule, and that's split and then each of those acts as a template for another half. And so we end up with two molecules, of DNA, and each of those two molecules is one half old DNA, one half new DNA, and we call that semi-conservative. Semi meaning half, conservative meaning that it's being saved, right? It's being conserved. So again, we call that semi-conservative. And I'll scroll down here on the notes. So let's start talking about the steps of DNA replication. And I apologize, the slides are a little messy in relation to the notes. So if you want to maybe pause and write down the notes first, it'll make it a little easier to talk through the slides. And so the first thing we need to talk about is that DNA doesn't actually copy itself. It's not the thing doing the copying. So DNA just serves as a template. It's essentially a book of code, and it says, hey, read me, copy me, share the good word, right? But the book's not going to read itself. And so enzymes, which are a specific type of protein, those, along with other proteins, do the actual work of replication. And so enzymes are a type of protein that either speed up reactions or help break things down. And so the example I've used a lot in class is lactose and lactase. So to explain what those are, we've said that sugars often end in the suffix ose, O-S-E. So things like glucose, fructose, sucrose, deoxyribose, or lactose. And so sugars end in ose, and enzymes, these proteins, end in ase, A-S-E. So if you see a word in biology that ends in A-S-E, chances are very high that it's an enzyme. And so lactose is a sugar. And so if you're lactose intolerant, it means that your body is not able to digest lactose that you acquire through dairy. Now, if you have, lactase, that is the enzyme that breaks down lactose. So if you're lactose intolerant, what happens is that your body actually doesn't produce either any or enough lactase to digest the lactose. So really here I just want to show you the relationship between the aces and the oses, right? So enzymes are things that do specific work. And so helicase is what we call the enzyme that unzips or opens the double helix. So helix, helicase. So helicase opens or unzips the DNA molecule, and that's the first thing that happens. So it's when replication starts. It unwinds and unzips the DNA. The second thing that happens is free-floating nucleotides form hydrogen bonds with the template strand. So remember that this is happening in the nucleus. DNA is in the nucleus. And so just like a cell has cytoplasm, the nucleus has what we call nucleoplasm, which is a very similar idea. And floating around in this nucleoplasm, or the nucleus is, that's a weird word, nucleus is, cytoplasm, are nucleotides, right? A's, T's, C's, and G's with sugar and phosphate. And what happens is now that the molecule has been unzipped, there are these bare exposed bases on either side. And so the nucleotides that are just free floating can detect, okay, these bases are now available to be paired with, and so they will be attracted to and then pair with their corresponding complementary base. So the nucleotides kind of float up and get paired with their correct base. So the A's pairs with the T's, the C's with the G's, and vice versa, right? That's the second thing that happens. From there, another enzyme called DNA polymerase. And before I talk about what it does, let's think about the name. Polymerase. So it literally is polymer enzyme. So remember, polymers are links of monomers, and nucleotides are the monomers of DNA. And so DNA polymerase is a group of enzymes that bonds the nucleotides together to form the double helix. So if these nucleotides have been added one at a time, the polymerase comes and then connects them into a strand of DNA, which really completes the double helix. And so the polymerase enzymes, they form covalent bonds between those individual nucleotides to make it a polymer. So it takes the monomers and links them with covalent bonds into a polymer. Now, there are a number of other enzymes that are involved in more specific ways in this process, but that's not really something that we need to worry about at this point. Those are things that you might worry about in college biology or AP biology if those are things that you choose to take. But if not, I don't think it's overwhelmingly important that we know those right now. So really those are the two that I want you to know, is that helicase unzips and unwinds, and that polymerase combines those nucleotides into a polymer, or the completed strand. And so for number 4, be a little patient here, it takes a second to get to it, but the next thing that happens is that you end up with these two new molecules of DNA. And remember that each of these is 1/2 old DNA, 1/2 new, and we call that semi-conservative. And so the reason that replication is fast and accurate is because it has multiple origins of replication. So it starts in multiple places at the same time. And so this is in eukaryotes specifically. But if we look here, and let's say this is our initial strand of DNA, we get this little bubble, or there's multiple places where this unzipping starts. So if DNA was a football field long, or imagine you had a zipper that was as long as a football field, and you had to unzip that whole thing in one direction, that would take a really long time. Well, DNA is comparatively much longer than a football field is, and it needs to unzip pretty quickly. So it unzips in multiple places at the same time, going both directions. And so they eventually run into each other, and you end up with two new strands. So this happens pretty quickly, and it happens pretty accurately because no one spot is doing too much of the work. And again, we call these origins of replication, which are the places where they start, or starting points. And so the final thing that happens is checking for errors. And I apologize that this isn't in red, but DNA has to be replicated perfectly or things are going to go wrong in the body or in the cell. So proteins could be misfolded or made incorrectly or not made at all. And so DNA polymerase, in addition to linking things together, they also can check for and then correct errors. And that's what I'd like you to have here for number 5. And so DNA polymerase both joins things, makes the polymer, but it also then checks for errors. It's kind of like its own spell check. And so if DNA copies incorrectly, we call that a mutation, or a change in the DNA code. And so this can cause all kinds of things to go wrong. It could, depending on the mutation, cause no problem, but it could also cause huge problems as things are misfolded or made too much or made not enough, or just any number of issues. And we'll talk more about mutations and the different kinds and the different implications later next unit. And so here, objective 9 asks you to review the structure of DNA. And now I know we haven't gone through objectives in order, so don't be worried that it's number 9. But I'm going to scroll down here, and I just want to talk through these rather than going through the slides. And we're not missing anything. We didn't go through these slides in class either, actually. So in class, what we did is to kind of pose these as questions and had you all fill them out. And so some things to remember, some key terms, some key concepts. DNA is a molecule that is a polymer, meaning it's made up of many monomers, and we call those monomers nucleotides. Now, those nucleotides have 3 parts. They have a phosphate, which we typically represent as a circle. They have deoxyribose sugar, which we typically, represent as a pentagon. Now you can just call that sugar, and that's okay. And then there are also the bases, or the nitrogen bases, or nitrogenous bases, whatever you want to call them. And those are what we see kind of on the inside. And those can be 4 different bases: A, T, C, and G. Adenine, thymine, guanine, and cytosine. Now, in a DNA molecule, we like to think of it as like a ladder. So there's the sides, or the backbone, and then there's the rungs of the ladder. And so the backbone is made of alternating sugars and phosphates, and sugar, phosphate over and over. And we call that the sugar phosphate backbone. The interior rungs, or the inner middle rungs of the ladder, are made up of the conjoined pairs of nitrogen bases, right? So maybe an A on the side and a T over here, or a G over here and a C over here. Those are the rungs of the ladder, or the middle thing that you climb up. Now, it's also important to remember the bonding. So just like a zipper is able to zip and unzip very easily without the sides breaking, the backbone is very strong and the rungs are easily separated or joined back together. And so strong covalent bonds are what hold the backbone together, but weak hydrogen bonds are what connect the two sides. And that's it. That's it for the notes for the entire DNA unit. So we had DNA structure and function. We had a little bit of history of the discovery of DNA structure. And then we have DNA replication. So that's it for all the new stuff this unit. I'll talk to you either online or in class. I hope you're doing well, and I'll see you soon.